Proteomics is the largest-scale study of proteins, according to Wikipedia. Wikipedia also explains the following about proteomics. Proteins are vital parts of living organisms with many functions, such as the formation of structural fibers of muscle tissue, enzymatic digestion of food, or synthesis and replication of DNA. In addition, other kinds of proteins include antibodies that protect an organism from infection and hormones that send important signals throughout the body. The proteome is the entire set of proteins produced or modified by an organism or system. Proteomics enables the identification of ever-increasing numbers of proteins. This varies with time and distinct requirements or stresses that a cell or organism undergoes. A quick advertisement before I continue on this topic. You can invest in my startup company with as little as $100 for supporting advancing AI, robotics, biotech, and nuclear fusion-powered aerospace tech. Visit robocentric.com/investors to invest in my startup. I have a multi-decade commitment to advancing AI, robotics, biotech, and nuclear fusion-powered aerospace tech. To learn more about my cause, check out my books, which are available at robocentric.com/checkout, Amazon, Apple Books, Spotify, and other online audiobook retailers. Now back to the main content. Proteomics is an interdisciplinary domain that has benefited greatly from the genetic information of various genome projects, including the Human Genome Project. It covers the exploration of proteomes from the overall level of protein composition, structure, and activity, and is an important component of functional genomics. Proteomics generally denotes the largest-scale experimental analysis of proteins and proteomes, but often refers specifically to protein purification and mass spectrometry, and mass spectrometry. Indeed, mass spectrometry is the most powerful method for analysis of proteomes, both in large samples composed of millions of cells and in single cells. Complexity of the problem in proteomics. After genomics and transcriptomics, proteomics is the next step in the study of biological systems. It is more complicated than genomics because of an organism's genome is more or less constant, whereas proteomes differ from cell to cell, and from time to time, distinct genes are expressed in different cell types, which means that even the basic set of proteins produced in a cell must be identified. In the past, this phenomenon was assessed by RNA analysis, which was found to lack correlation with protein content. It is now known that mRNA is not always translated into protein, and the amount of protein produced for a given amount of mRNA depends on the gene it is transcribed from and on the cell's physiological state. Proteomics confirms the presence of the protein and provides a direct measure of its quantity. Post-translational modifications. Not only does the translation from mRNA cause differences, but many proteins also are subjected to a wide variety of chemical modifications after translation. The most common and widely studied post-translational modifications include phosphorylation. I'll check pronunciation later. I didn't have the time to check all the bioscience terms. We just schedule. Phosphorylation and glycosylation. Many of these translational modifications are critical to the protein's function. Phosphorylation. One such modification is phosphorylation, which happens to many enzymes and structural proteins in the process of cell signaling. The addition of phosphate to particular amino acids, most commonly serine and threonine. I'll check the pronunciation later. Mediated by serine threonine kinases, or more rarely tyrosine mediated by tyrosine kinase causes a protein to become a target for binding or interacting with a distinct set of other proteins that recognize the phosphorylated domain. Because protein phosphorylation is one of the most studied protein modifications, many proteomic efforts are geared to determining the set of phosphorylated proteins in a particular cell or tissue type under particular circumstances. This alerts the scientist to the signaling pathways that may be active in that instance. Ubiquitination. Ubiquitin is a small protein that may be affixed to certain protein substrates by enzymes called E3 ubiquitin ligases. Determining which proteins are polyubiquitinated helps us understand how protein pathways are regulated. That is, therefore, an additional legitimate proteomics study. Similarly, once a researcher determines which substrates are ubiquitinated by each ligase. I check the pronunciation later. I know I shouldn't be lazy, but I'm on an impossible schedule here, doing other things and then producing these content to just keep practicing my communication or mass communication skills, and also to my biotech research and other researches. So I promise I'll do a pronunciation check later and correct my pronunciations after producing these content. Determining the set of ligases expressed in a particular cell type is helpful. Additional modifications, in addition to phosphorylation and ubiquitination, proteins may be subjected to, among others, methylation, acetylation, glycosylation, oxidation, and nitrosylation. Some proteins undergo all these modifications, often in time-dependent combinations. This illustrates the potential complexity of studying protein structure and function. Distinct proteins are made under distinct settings. A cell may make different sets of proteins at different times or under different conditions. For example, during development, cellular differentiation, cell cycle, or carcinogenesis, further increasing proteome complexity. As mentioned, most proteins are able to undergo a wide range of post-translational modifications. Therefore, a proteomics study may become complex very quickly, even if the topic of study is restricted. In more ambitious settings, such as when a biomarker for a specific cancer subtype is sought, the proteomics scientist might elect to study multiple blood serum samples from multiple cancer patients to minimize confounding factors and account for experimental noise. Thus, complicated experimental designs are sometimes necessary to account for the dynamic complexity of the proteome. Robocentric will definitely get into proteomics, biotech, hardware, software development, and marketing endeavors, and radically contribute to advancing the field of proteomics through developing and marketing each proteomics, biotech, hardware, and software. So that's one of the things that Robocentric will commit itself to for over a very long term, like multiple decades, not just short term. Okay, limitations of genomics and proteomics studies. Proteomics gives a different level of understanding than genomics for many reasons. The level of transcription of a gene gives only a rough estimate of each level of translation into a protein. mRNA produced in abundance may be degraded rapidly or translated inefficiently, resulting in a small amount of protein. As mentioned above, many proteins experience post-translational modifications that profoundly affect their activities. For example, some proteins are not active until they become phosphorylated. Methods such as proteomics and glycoproteomics are used to study post-translational modifications. Many transcripts give rise to more than one protein, though alternative splicing or alternative post-translational through alternative splicing or alternative post-translational modifications. Many proteins form complexes with other proteins or RNA molecules and only function in the presence of these other molecules. The protein degradation rate plays an important role in protein content. Reproducibility. One major factor affecting reproducibility in proteomics experiment is the simultaneous elution of many more peptides than mass spectrometers can measure. This causes stochastic differences between experiments due to data-dependent acquisition of tryptic peptides. Although early large-scale shotgun proteomics analysis showed considerable variability between laboratories, presumably due in part to technical and experimental differences between laboratories, reproducibility has been improved in more recent mass spectrometry analysis, particularly on the protein level. Notably, targeted proteomics shows increased reproducibility and repeatability compared to shotgun methods, although at the expense of data density and effectiveness. Data quality. Proteomic analysis is highly amenable to automation, and large data sets are created, which are processed by software algorithms. Filter parameters are used to reduce the number of false hits, but they cannot be completely eliminated. Scientists have expressed the need for awareness that proteomics experiments should adhere to the criteria of analytical chemistry, sufficient data quality, sanity check, validation. Methods of studying proteins include protein detection and antibodies, immunoassays, antibody-free protein detection with a detection method and separation method, hybrid technologies, current research methodologies, high-throughput proteomic technologies with mass spectrometry and protein profiling, affinity proteomics, protein chips, reverse-phased protein microarrays, and protein detection via bioorthogonal chemistry. Practical applications of proteomics include the new drug discovery, interaction proteomics, and protein networks, expression proteomics, biomarkers, proteogenomics, and structural proteomics. Bioinformatics for proteomics, proteome informatics, include protein identification, protein structure, post-translational modifications, computational methods in studying protein biomarkers. Emerging trends in proteomics include the systems biology and human plasma proteome. I'll plan and execute my own proteomics biotech research and development in sensor, scanner, modeler, designer, synthesizer, and place of biotech categories for advancing medical biotech, human longevity biotech, and other types of biotechnologies in the decades to come. You can invest in my startup company with as little as $100 for supporting advancing AI, robotics, biotech, and nuclear fusion-powered outer space tech. Visit robocentric.com/investors to invest in my startup. My books on advancing AI, robotics, biotech, and nuclear fusion-powered outer space tech are available at robocentric.com/checkout, Amazon, Apple Books, Spotify, Google Play Store, and other online audio retailers. I am Alan Young. I'm an Asian American man who focuses on advancing AI, robotics, human longevity biotech, and nuclear fusion-powered outer space tech. Message me or call me anytime on the social media if you'd like to discuss investing in my high-tech startup.